Okay. Also willkommen allerseits. Wir sind 13, wir sind ein Quorum sozusagen oder 50% sind da. Guten Morgen, guten Nachmittag, guten Mittag, guten Abend, wo immer ihr herkommt. Mein Name ist Alexandra Lieben, ich bin Chapter Head von Austria Pacific South, unserem Chapter hier. Und abgesehen davon bin ich Deputy Director vom UCLA Berkeley Center für International Relations. Ich freue mich, euch heute zu begrüßen und freue mich sehr über den heutigen Vortrag und euch Daniela Weiskopf vorzustellen und die Arbeit, die sie macht, die wirklich sehr spannend ist und sehr zeitgemäß, würde ich sagen. Ein paar Worte über Daniela und dann spricht Daniela ungefähr 40 Minuten lang. Ich setze ihre Bio auch in den Chat hinein für alle, die zu spät gekommen sind und es nicht mehr gehört haben. Aber ich bitte euch dann, während ihr zuhört, überlegt euch eure Fragen und gebt die auch in den Chat hinein, damit wir es dann, also wir machen 40 Minuten Vortrag und dann ungefähr 15 Minuten Q&A nachher. Also, Daniela, willkommen. Willkommen zu Austria und zum heutigen Vortrag. Daniela ist Research Assistant Professor beim Center for Infectious Disease und Vaccine Research im La Jolla Institute for Immunology. Abgesehen von allem anderen ist sie auch der Recipient vom 2021 Austria Junior Principal Investigator Award. Congratulations again. Daniela devoted her career to understanding the T-cell response to viral pathogens and spent the last 16 years studying infectious viruses relevant to human health and disease. In 2009, she received a PhD in immunology from Innsbruck Medical University in Austria. Following her PhD, she obtained postdoctoral training at the La Jolla Institute for Immunology, where her efforts were dedicated to characterizing human dengue virus-specific CD8 plus and CD4 plus T-cell responses. She also studied Zika and chikungunya virus-specific T-cell responses and became interested in the effects of pre-existing immunity against dengue virus to subsequent Zika virus infection. As a research assistant professor in the Center for Infectious Diseases and Vaccine Research at the La Jolla Institute for Immunology, she has focused most recently and very aptly on the characterization of the SARS-CoV-2 specific T-cell responses to better understand adaptive immune responses to SARS-CoV-2, which matters for vaccine development, for interpreting disease pathogenesis, and for the calibration of future pandemic control measures as we face variants down the line. With this, join me in welcoming Daniela. Daniela, the floor is yours.
Thank you so much, Alexandra. Yes, I'm very excited to be here and present what I have been doing the last 2 years to this audience. And as you heard, I will talk today about the immune responses that are induced by COVID-19 vaccines. And that is a question that is on a lot of people's minds, as we have mostly all of us have been receiving a vaccine. So I'm just gonna present to you what we have learned in the last 2 years about that. As you heard, if you could have not already guessed from my Twitter handle, I'm from Innsbruck, 20. And this is also where my entire career before I came here to Southern California has taken place. I have studied at the University of Innsbruck. I have gotten my diploma in microbiology. I have then done my PhD thesis at the Medical University of Innsbruck. And after 10 years, I came to the La Jolla Institute for Immunology to perform my postdoc. So I've basically switched the snowboard to the surfboard, but otherwise it's pretty similar in what I like to do outside of the lab. And what have been connecting also, aside of that, is really I have always been interested in viruses. And that was from my diploma thesis, from my microbiology days, all the way to now. I'm interested in how our immune system recognizes viruses. And as you can see on the right side, I'm excited for my parents, was also picked up by the Jolla Tageszeitung. So this is something that has always been near and dear to my heart. And I have been studying, as you also heard from Alexandra, mostly other emerging infectious viruses, such as dengue virus, Zika virus, chikungunya virus, and yellow fever. And of course, we know that this is something that we need to be like prepared for, and we need to understand what happens if a new virus is emerging. And basically, we all know that 2 years ago, we did all encounter the next emerging infectious virus. This was SARS-CoV-2. Because we have been spending so much time to establish methods, to like understand like what samples do we need to study, like what do we need to look at, what's important. We were like up and running very early in this pandemic because we already had like everything in place, because I have been studying like emerging infectious viruses like all my career basically. So that's what we have been doing. And then one thing I want to point out is like also like something that we have also been establishing when we think about other emerging infectious viruses, and which is also like been doing here. So the way I'm thinking about viral infections in human, I think there's somebody in the background. So the way I'm thinking about like studying immune responses against emerging infectious viruses is always like threefold. I want to know, what is the natural immunity look like? That means people that have gotten the virus and have recovered from it. And why do I'm interested in that? Because like if you do not know what like the natural infection with the virus looks like, you cannot benchmark the vaccine response. So you don't know if you make a vaccine, if response is like better, worse, or just as good as a natural infection. That's important because like you ideally want to have like a response that is like, you know, equally as good as natural immunity or better, without like the added risk of like having infection. And you can already see like within this natural immunity pool, there is like a big variety. So when you study human populations, like you have typically all kinds of spectrum. Like you have people from like that never even know they have the infection, so asymptomatic infections, people that have mild disease, and people that have severe disease. And that is also like brings us straight to like our next corner here. So I'm also interested in people that have the acute disease. So not only like when they are in the hospital, like how do they look like? How does the immune response look like? Is there anything different that can tell me like who will have like mild disease, who will end up with severe disease? And that's also important to understand, to put them in context when you like study vaccine induced responses. So this is a graph that is still like from their studies when I'm studying flaviviruses. Of course, when you study human immunology, like this is not mice. So a mouse has like one infection at a certain time point, and then you know with what strain this mouse has been infected and at what time point, and that's it. If you study humans, it's not like an isolated event. So everybody of us has like, you know, had multiple infections with all kinds of different viruses. We do not know how they influence each other. And that has been a very big question in my field, what I studied before, and I still do, flaviviruses. So you have had the infection with dengue virus before, and then you get Zika infection. Is there any influence or interference of the immune response to one virus with the other? And you will also hear like in a little bit, the very same question came up in SARS-CoV-2, when we quickly realized that there's people that have immune response against this virus without having seen them. And that is also relevant for vaccine studies. And then I will gonna go show you all the data now. So that's basically my background, where I was coming from. And now I'm gonna go and show you what I've been doing the last 2 years and study COVID-19 vaccines. So the last thing I want to point out, which is also a general topic, is that we always look at all three different kinds of the adaptive immune response. So we always study antibodies, we study CD4 T cells and CD8 T cells. Why do we always study all three things? Because like they all tell you very different things. So antibody response is usually something that is needed when you get infected. This is the first line of defense. You will like have antibodies that can attack the virus and then can be neutralized. Once the virus is inside a cell, antibodies cannot do anything anymore. So because like the antibody cannot go inside a cell. What you need then is like the cellular immune response that can recognize infected cells and then can actually like go ahead and like eliminate virus infected cells where antibodies wouldn't be able to do anything. So, and likewise, CD4 and CD8 T cells take a while to be induced, be activated. So that's when you need antibodies first. So all of these things are important to have a successful immune response. And depending on the virus, one is more important than the other one. But in order to find that out, you need to look at all three of these things and to establish what might be the correlate that of protection. As I pointed out, and I'm quickly going to show one slide here. We need to understand how all of these parameters look in natural infection, because otherwise we cannot and extrapolate what does this mean for vaccine response. So this is one graph that we have been studying people that have had mild disease and have recovered. So we consider this a successful immune response because you have been infected, you have recovered, and you basically, your success is measured by you being alive after infection. So we have shown here all the different parameters that I've been just telling you about. So on the left side, all in blue, is antibody responses. We look at different kinds of antibody responses because they also have different functions. IgG neutralizing antibody titers, which is the one that is the functional response that can actually neutralize viruses. IgG is antibodies that are typically associated with mucosal immunity, which is particularly important for respiratory viruses, the one SARS-CoV-2 we have been just dealing with right now. And then all the way on the right here is cellular immune response. So that is on the top, the Spike-specific memory B cells. So memory B cells are the cells that do make antibodies. So that's important because even when antibodies decline, as long as you have memory cells, that means you can quickly release more antibodies if needed. As we also see in the next graph, CD4 and CD8 T responses, and you can see that for all of these parameters that I've been showing you is there's a broad heterogeneity. So there are some people that respond very strongly, some people that respond poorly, some people that have after 6 months, 6 to 8 months, you can detect very well all parameters of the immune response, and some that you lose that. So about 10% of individuals have low levels of immune response. So what we don't know yet is who are these 10%? And when people say I don't need a vaccine, I'm fine because I've been infected, you don't know if you're part of the 90% of the part of the 10%, because if you're part of the 10%, you do not have any immune memory anymore. It's basically you have never seen the virus again. And this is just to summarize what I've been showing you. When you look at the infection, natural infection after 1 month, the majority of people have 3 or more of these immune parameters very readily detectable, and about 90% still have 2 or more of these any of these parameters detectable. But as I said, 10% do not have anything detectable anymore. So and we still don't know what's different with people that do have that and do that have not. So now that we understood pretty well how natural infection looks like, we were interested in what does the exact same thing look like in vaccine response. So this is a study that has been performed with the mRNA 1273 vaccine, which is the one that is mostly referred to as the Moderna vaccine. The Moderna vaccine has been given at 2 different doses, 28 days apart. And this study has been performed on phase 2 clinical samples, which were back when they tested what is the perfect dose. So this is 25 microgram of this vaccine has been given twice. We all know by now the dose that has been given is 100 micrograms, 4 times as much. But we still nevertheless wanted to understand how does this immune response look like, even though the vaccine is given as a quarter of the dose. We also want to point out we had access to different age groups because there was always the concern that the vaccine might maybe work in the young cohort, but not in the elderly. So these age groups were going from 18 to 55, 56 to 70, and over 70. So we had representative samples from all of these different age groups to understand if there is anything different in any of these age groups. And then on the right side, it just shows you the different visits that we've been collecting. We have been collecting 4 different samples from each of these vaccine trial participants. Importantly, the visit 1 at enrollment is important because you need to understand what the immune response or what the memory, the immune state is of a person before it got the vaccine. because as I told you before, like humans are not like mice. They have been during their life, like, you know, are exposed to multiple different infections. So you need to understand what the immune status is before a person gets the vaccine. So then you can put it into context when you collect samples afterwards. So we then collected after the 2 different doses, 2 weeks after the first immunization, 2 weeks after the second immunization. And then importantly, because like we also wanted to know how long of immune memory can be established by a vaccine response. And that's important because you need to understand how the immune response looks like after 6 months, 6 to 9 months, because if you look too close, then I'm sure you all heard about, like, you know, the people that said like, oh, the immune response is waning, like, you know, it's declining very fast. And that is if you measure too early, 1 to 3 months after infection or vaccination, you naturally will see a decline above any of these immune parameters. That's just how the immune system works. So if you look too close, you will always see a decline, because that's just like the immune system contracting from effector phase to the memory phase. So it's important you have a sample collected like far out, 6 to 9 months after infection or vaccination, to fully understand like what's the level of immune memory that is being induced by this, in this case, the vaccine. That's why 6 months after second immunization was our last time point. So quickly going through some of the results. So as you can see here, we also measured antibodies just like for the natural infection. And you can see here, after 6 months, we see antibody parameters like in 100% of the people. So that's higher than what we've seen after natural infection. So all of these people that have been followed for 6 months still have antibodies after like they received the 2 doses of the vaccine. And not so surprising, maybe that the highest response was measured after the second dose. So this is also showing you the reason why we need 2 doses, because like it really lifts the immune response, the responders from like 86 in this case to 100%. So good news, antibody responses are induced in all of this cohort. And as I pointed out in the beginning, also like old people were in this cohort, and everybody made antibody responses and was able to maintain them up to 7 months. What about the T cells? I'm showing CD4 T cells here. And we also see a similar picture. So again, the strongest immune response was like CD4 T cell response was induced after the 2 doses of vaccine. So also here it's beneficial to get 2 doses of the vaccine, because you make sure that everybody has CD4 T cells induced. When you like follow the response after like 6 to 7 months, you see that only 1 of the people dropped under the like detectable line. So again, 97% of the people still have detectable response in the memory phase after 6 months after the second dose. So again, that's good news, because like that means like the vaccine induces very durable immune responses, cellular and also antibody responses. We have measured a similar thing for CD8 T cells and B cells. I'm not showing this right here right now. So on overall, the summary is like, how has this vaccine response been performing? So again, this was 2 different shots, two different doses, 25 micrograms, so 1/4 of the dose that's given to the majority of people. If you compare it to natural immunity, it's equally, so it's the response magnitude is similar to what we've seen after natural immunity. And that's important because like with only half of the dose, you have like just the same response. With only 1/4 of the dose, you have just a similar response as if you would have been recovered from natural infection. So we then compared it, of course, to the 100 microgram, because this is the dose that most of us have received. And you can see that if you get 100 microgram of the vaccine, the antibody response is even stronger, and the CD4 T cells is even stronger. We didn't see a difference in the CD8 T cell response. We were then interested in if there's any difference in the age groups, and we only saw that there is some antibody responses are declined in the very old age groups, so 70 and higher, but there was no difference in CD4 T cells and no difference in CD8 T cells. So that's important, because that means like while you have like might have a lower antibody response, you still have just as good induction of the cellular immune response, and that's important because they could really like have a difference in like how severe of an infection you could get. And I follow up on this in a second. So I pointed out that like I have always been interested in interaction in the human host between like infections with viruses that are closely related and people have seen before. And we noticed early on in this pandemic that we see responses in about 50% of the people where we know they have never seen the virus. And how do we know that? Because we have liquid nitrogen tanks that we have stored samples from like 2015, 16, 17, like long before the virus, this virus, SARS-CoV-2 virus, was actually like present and circulating. So if you test blood samples from people that have you collected way before they have seen, before this virus was circulating, and of course we also like made sure that serology confirmed like our assumption that they have never seen the virus, still we see in about 50% of the people that they still have CD4 T cells that recognize this virus. What we didn't know, and then where's this like cross-reactivity coming from? We were speculating it could come from common cold coronaviruses because they are the closely related cousins of SARS-CoV-2, but we didn't know at this point. And what we also didn't know, does it have any difference? Like, does it have any effect on subsequent infection or vaccination? Are you better off if you have these cross-reactive pre-existing immunity? Are you like worse off? Does it hurt you if you have this? Or does it maybe not make a difference at all? So that's something that we were interested in. And we, in the first attempt, we're trying to figure out like where this cross-reactivity is actually coming from. And we have demonstrated in this publication, this is actually in fact because people have previously been exposed to common cold coronaviruses. And this is just to show, like, if you look at the original SARS-CoV-2 peptide, and then you give the same blood sample, the corresponding peptides from common cold viruses. And if you are not aware, there is 4 different viruses, common cold coronaviruses, that are relevant for human health and disease. Mostly children get infected with these throughout their lifetime, all 4 of these, but also like, you know, parents, grandparents that like take care of children, so like are constantly exposed to that. So we could show that like these parts of the virus that are recognized for SARS-CoV-2, these blood samples also recognized samples from like common cold viruses. So in this case, is an example for like one of these virus segments that is recognized in 2 other common cold. We also saw the cases were like all like very conserved against all of the other coronaviruses. So you can see here that the original peptide is shown here in black, but like the corresponding peptides from common cold are even recognized stronger than the one, which is indicative that this was like the common cold infection was the first infection. So this was important because that answered the first question, like, yes, there is pre-existing immune memory in about half of the people, and this is derived from common cold coronaviruses, which all of us are infected. And this is actually also the publication I then received the Astronaut Junior PI Award. So this was exciting because like I was very excited that this was recognized by my home country. So it was a good experience. And as I said, like this publication only demonstrated that where this cross-reactivity is coming from. We still didn't know the second part of the question. Well, does it even have like an effect? Are you better off or are you worse off? And that's what we were then interested in next, answering the next question. So we had the perfect cohort because we had like this vaccine cohort where we have collected samples before they have received the vaccine. So that allowed us to determine if our vaccine cohort actually has people that have common cold, that have cross-reactive pre-existing immunity, and people that have not. And just as we would have expected, our cohort almost fell into like perfect 50-50. So you can see here, all the blue dots show people that do have pre-existing immunity, and then there is the open dots that do not have pre-existing immunity. So now, because we had like a perfect split, near to perfect split of our cohort, we could ask, well, is there any difference in subsequent vaccination? And we split all of our analysis based on these 2 different groups. So pre-existing immunity in blue and non-pre-existing immunity in the open bars. And you can see shown here for CD4 T cells, that not only do they respond much stronger, but also the kinetic response is much faster, and the immune memory, CD4 T cell memory in people that had pre-existing immune memory in the beginning was like significantly higher, even like 6 to 7 months after the infection. So that was very encouraging and interesting because like that only for the first time, like now demonstrated, not only do we have this fixed immunity, we know where it's coming from, but it also makes a difference. So people do have stronger and faster CD4 T cell response. It didn't have any effect in CD8 T response, that's why I'm not showing it here. And it had like some effect on the functionality of CD4 T cell response, which is shown here on the right. So what I have mentioned before is like it's always important to look at all these parameters like in concert. So you need to look at the same person, like the antibody response, T cell response, to fully understand the picture. And the hypothesis here was like if you have like strong induction, faster induction, and more prolonged induction of CD4 T response, this has to have an effect on antibody responses, because we know one function of the CD4 T response is to provide T cell help to antibodies. So we looked at antibody responses subsequently here, and in the exact same donors again split based on like if they had pre-existing CD4 T cell memory or not. We see a faster induction of antibody responses, as you can see here. On the second time point we looked at, it's significantly higher responses. Also, like if you look at the functional response, which is very shown here on the right for neutralizing titers, you can see that the group that had pre-existing CD4 T cell response, and that's important. So we measure here antibodies, but like having pre-existing CD4 T cell responses made a difference in the immune memory. So like 7 months after vaccination, you are better off if you had like pre-existing T cells before vaccination. So we are not the only people that have seen that subsequently. Like, so after we have published this, like there's been more and more interest in like, could that explain why people see such a big difference in symptomatic and asymptomatic cases? We all know that there is groups of people that have not really like been hit so hard by like by a severe disease. And one question, like what all of this study like brought up now is like, well, could this pre-existing immunity have an effect on that? And so we showed that for vaccination, but like after then, like also people showed that for like natural infection settings. So healthcare workers that are constantly exposed to like, people basically coughing in their face. So like they've been constantly exposed to coronaviruses, common cold coronaviruses, other respiratory viruses, do have a stronger T cell response against SARS-CoV-2, even though I haven't never seen this virus. There's been other studies around from like the United Kingdom or Germany, also from like Singapore, that just demonstrated that people that do have this cross-reactive immune response do respond stronger and faster after vaccination and or natural infection. So that is basically that we, at the point when we like this, when we convinced ourselves, okay, natural infection and our immune system is very capable of like recognizing this virus and is very capable of forming memory response, either natural infection or vaccination. That's when like all the variants started occurring. So we were interested in like, well, is this a game changer? Like, does everything that we have learned so far change this? So we were interested in asking the question like, well, okay, so now you have been vaccinated against the ancestral sequence, you have been exposed to the ancestral sequence, but now you see any of these variants, does the outcome change? So we designed basically peptide pools, which is our like tool, so how we can measure like virus-specific T cell responses against all of these different variants. And you can see it goes from like alpha to Omicron, which we hope is the last one, but we're not sure. There might be more and more to be added here. And asking the exact same question, like, okay, do T cells that have been primed by the original sequence or by the original vaccine still recognize any of these different variants? And the answer is yes, good news. So it doesn't matter what vaccine you have been receiving. So we have been looking at people that have received the mRNA vaccine, what I've been showing you right now, that have received the Pfizer vaccine, or that have received the Johnson & Johnson vaccine, which is very similar to what people in Europe get AstraZeneca vaccine. So it's post-vaccine designed with a viral vector. So we had access to these three different kinds of vaccinees. And we were asking the question, okay, so you have been primed against the original ancestral sequence. Do you still recognize any of these variants? And the answer is yes, good news. It doesn't matter what variant we probe these memory. And as I pointed out, importantly, have been primed by the original sequence. all of them still recognize any of these variants equally good. In some cases better than the original one, but on average, like basically just as good. And that was the case for CD4 T cells and also for CD8 T cells. So that's good news, because like as you might be aware, like that same is not true for antibodies. So antibodies like not always recognize all the variants equally good. But as I also just convinced you, I hope, is that having T cells that recognize this virus can make a difference, because we have demonstrated having pre-existing T cells can make a difference in your vaccine response, can make a difference in subsequent infection. So even maybe your antibody response against any of these variants might be different, you still have your second line of defense that just recognizes these variants equally good. So I have not shown you on the very first slide Omicron, but the same thing is true for Omicron. And the same thing is also true of the memory point. So if you look at this graph I've just shown you, this has been all measured like 30 days after vaccination. So this is basically showing you, yes, shortly after vaccination, you recognize all variants. And the next question was, as always, like, what if you measure now the memory response? And the response was just equally the same. So it doesn't matter what variant you look at, even 5 to 6 months after vaccination, you still recognize your T cells, still recognize all of these variants with equal magnitude. So then, after we have addressed all of this, we were interested in what if you do mix and match, because what I've been showing you so far is like people that have been vaccinated with like a certain vaccine with the same vaccine twice. What if you now go and like have received a different vaccine at the first dose, then you have then received a different vaccine for your second dose? And that is something that I connected back to my alma mater. Professor Van Leer and Dr. Janine Kimpel and Dr. Sultan Banki have been running in the district of Schwab, which is here like pointed as number 6, a study that has looked at people that have received AstraZeneca before and then as a second shot gotten the Pfizer vaccine, and compared it to like people that have received the Pfizer and Pfizer. And I don't know how familiar you were with what was happening, but you remember that like very early on, the South African variant was like circulating heavily in the district of Schwab's. So that is why there's a lot of people early on that have been vaccinated with like the Pfizer vaccine twice. And that's why also we were interested in like, okay, so if the AstraZeneca immune response is not good enough to like protect against variants, or like not as good protect against variants, is a difference if you then have like a different vaccine as a second shot, something that we refer to as heterologous vaccination. That means your first and second dose was different. And as I just pointed out, like these are the 3 groups that we have been looking at too. There's one group that have received for the first and for the second dose the Pfizer vaccine. There is people that have received for the first and for the second dose the AstraZeneca vaccine. And then we were particularly interesting, like what does it look like if people had gotten the AstraZeneca vaccine first and then gotten the Pfizer vaccine as a second dose? Is their immune response same, different, or like what does it look like? So if you look at the antibody response, and that's again, the 3 groups that I've been just introducing to you. So the blue one here shows people that have received AstraZeneca, followed by AstraZeneca. The purple one is AstraZeneca and then Pfizer, which is the group we were really interested in. And then the control group was people that have received Pfizer and Pfizer at the same time. And you can see that the antibody response, the spike-specific antibody response was much stronger in the people that we have heterologous vaccinated if you compare it to like people that have received AstraZeneca and AstraZeneca. As my lab is interested in T cells, that's the next thing that we were asking. And we were interested in asking the same question. So is there a difference in the CD4 T cell response or CD8 T cell response, depending if these vaccines have been like homologous or heterologous primed? And the answer is yes. So if you again look at the blue group here, you can see the immune response, the CD4 T cell response induced by AstraZeneca, and it's lower, significantly lower than if you have received AstraZeneca as a first and Pfizer as a second shot. Again, this is not different if people that have received Pfizer and Pfizer for both of their doses. So by giving the second shot a heterologous vaccine, you raise the immune response, even though like the first shot was like a viral vector vaccine. So that is true for CD4 T cell responses, but interestingly, there was no difference in CD8 T cell responses. So that's something that we keep seeing over and over and over, like CD4 T cells seem to be much more differentiating responses, immune responses against this virus, because CD4 T cells, as I showed you before, CD8 T cells, we didn't see a difference in, like, depending on the age, we didn't see a difference depending on here now different prime boosts. We didn't see much a difference depending if you have like a high dose, a low dose, or natural infection. So CD8 T cells seem to be a lot less affected by this virus. So the differences really seem to be like breaking down to CD4 T cells. And that's important because I also shown you like that the magnitude and the functionality of CD4 T cell responses does directly influence like the antibodies you can like make against any of these viruses. So again, here we showed not even if you have like a homologous vaccination regime, but you have like a heterologous, so for different ones, CD4 T cells and antibody responses are influenced. So the other thing that we also always look at, and I have touched briefly on the side, is like we're not always only looking at the magnitude of response. So meaning like you can just induce more of anything. So we're also interested in the functionality. So like if you study antibodies, you will not only measure the titers, you will also measure neutralizing titers, meaning like how good can an antibody neutralize any of these viruses. For the T cell part, we are equally interested in like, okay, it's just the magnitude different, but the functionality of T cells might be the same, because then it's just a numbers game, you have more of. But does it also like influence the function? So this is the question we've been asking here. And again, we do see that the functionality, not only the magnitude, but also the functionality. So the way we measure functionality is based, we look at how different effector molecules this antigen virus specific cell can produce. And that's how we can calculate like the level of functionality. So you can see the more of like the pie chart is like less than dark blue. That means like they have more than 2 functions in response to this heterologous vaccine response. So that was true for CD4 T responses. And I just showed you a slide before that to be not only see stronger immune responses, but now I'm showing you also like more functional spike-specific CD4 T cell responses. And very interestingly, I just showed you that CD8 T cells, the magnitude is not affected at all by this, but so is the multifunctionality. So you do not make more of CD8 T responses if you have like AstraZeneca followed by Pfizer, but you make a different functionality of the CD8 T responses. So that's very interesting because it's not always like just the amount that counts, you know, it doesn't always mean like, you know, stronger immune response is better. In this case, it shows that maybe the magnitude is not different, but clearly both of these assays measure that there's a different function of CD4 and CD8 T response and that virus specific induced by these heterologous vaccine regime. So to conclude this part, I can show you that this heterologous vaccination uses higher spike-specific antibodies. I showed you that it induces stronger magnitude and also quality of spike-specific CD4 T cell responses. And also, like I showed you that while this heterologous vaccine regime does not induce a strong CD8 response, but it changes the quality, the functionality of these CD4 T cell responses. So this is basically summarizing, and I wanted to show you everything that we have been learning so far over the last 2 years about this virus in natural infection, also vaccination. And I've pointed out before, it's important to measure all these different parameters because from everything we learned so far, we really do believe they have a different functionality and role. So if you're looking for protection against detectable infection, you need strong antibody responses because that's the ones that only antibodies can provide something that people refer to as sterile immunity. CD4 T cells cannot do that. So because I'm sure you heard about people until like I got vaccinated and still got infected. Yes, well, but the question is not if you still got infected. The question is, have you been like, you know, in the hospital or not? So, because if you are looking at the question, can vaccines protect against hospitalizations and deaths? You need an equally equally strengths of all these immune parameters. You need like antibodies, CD4 or CD8 or a CD or a B cell plus that can make these antibodies. We still don't know to this point like what is the most important one, and also what we still don't know is like what correlates with the other one. So does having high antibodies always mean you have high CD4 T cells? That's not what we have seen. So it's not you cannot measure your antibody levels and then assume that all your other immune parameters are just equally as good. So that's something that needs to be kept in mind, and by carefully dissecting this immune responses over this last 2 years against this virus, we have learned a lot. And we have, of course, because that's why it's research, open a lot more questions that will keep us busy going forward. And with that, I'd like to thank all the people that have been involved in this. You can see all of the papers that have been receiving the award have been performed by Jose Matias, which is a postdoc here in the lab. And I do work closely with Alex Sette and Shane Crotty here at the Institute. And by combining all of our expertise, we have really been able to look at all these different immune parameters. And with that, I'd like to thank all of you for your attention, and I'm happy to take any questions if there is any.
And I would like to invite everybody to unmute yourselves and turn on your cameras, or rather turn on the cameras, don't unmute yourself. Daniela, I have a quick question. That means no more flu shots? So we strengthen our immune system? No.
No. So the flu shot is very different because the virus is mutating much faster, right? So that's why you always need an adjusted one, and that has been a discussion here for this the COVID vaccines too, right? Do we need to every 6 months get a booster now? And there's different scenarios the way I'm thinking of it. So I think if you're immunocompromised, yes, you should get a booster probably every 6 months, because we just don't know how high the wall needs to be to keep the virus at bay, right? We don't know how much high level immune response you need to be when you didn't have a good response in the beginning. The other consideration could be if there is more different variants appearing that will not be able to be recognized by the immune system. Yes, we would need to have a variant specific booster. If you just need to induce the level of immunity, it looks like you can just use the same ones we had so far, because boosting increases the antibody level. It's also possible that, what we see, because it doesn't look like immune memory is falling off the cliff, but until we know what is the level that we need, we still need to consider boosters or not. So that's what I'm saying, a lot more questions to answer.
So quick follow-up question on the booster issue is should we then, following your research results, findings, should we get a booster from another vaccine than the one we originally had?
Yeah, that's a good question, because it does look, so this was comparing completely different vaccine manufacturers, right? So the first one was a viral vector and the other one is the mRNA technology. We don't know if the same effect would happen if you get Pfizer first and then Moderna. So we don't know that. So that's something that needs to be, because they're just the same technology. Certainly, if you have received AstraZeneca or J&J, I think you're better off getting an mRNA vaccine booster on top of that. Or there is also I think about to be licensed in Austria, or already is, Novavax is a protein vaccine. That's another vector. So it's basically, instead of having an mRNA vaccine, you have the entire protein. So that we don't know how that looks like if you mix and match. So yeah, remains to be seen. But yeah, certainly if you received AstraZeneca, then that definitely makes a difference if you mix and match.
That was a really fascinating talk. Thank you so very much. Questions? Anybody? And we would like to see your faces, because it's very hard to have a conversation when we don't see anybody. That is the other thing. There we go. Dietrich, you have a question?
No, fantastic talk.
Interesting. Daniela, follow-up question. I come out of public policy, so my question is always policy related, or my thinking. Yeah, and what, based on your findings, what is your advice to public health agencies?
Yeah, that's a whole different topic, because if there has always not been agreement on what science says and public policy says, right? And there's certainly also been always maybe a lack of communication, or how things have been portrayed, because we as scientists always knew that this vaccines is never gonna provide sterile immunity. But I don't think that has been communicated good enough to the public, because I know a lot of people were disappointed when they got got infected, even though they said like, well, I got all the vaccines and the booster, and I'm still getting infected. So I think there is a lot of work for you, policymakers, that can like, decide what's better for like the community as a whole. So yeah.
So I actually
I leave that in your part. Yeah.
Let me follow up on that, because I'm neither, in immunology nor public policy. But where do you see society going now? With some societies, basically saying, oh, things are over, we're back to normal. Others say, oh my God, something's getting worse again. We should read Austrian news. This is what they think they see there. And other areas where, we still have, I had the opportunity to talk with Florian Klammer last week or 2 weeks ago at the embassy, and he talked to me about, his efforts also to bring vaccines to underserved areas in the world. Are we talking about completely, parallel societies happening? And
I mean, you're basically already nailed it with your point. As long as we don't vaccinate like the world, there will always be like, opportunities for the virus to mutate. Because how can the virus mutate? I can provide the host for the virus and allow it to mutate. You can do that as long as we all are letting virus infect people. And that is also, if we don't vaccinate, big parts of the world, the virus will always have opportunity to mutate. So that being said, we don't know, right? So Omicron seems to be mitigated pretty well by the booster. There might be another variant coming up, which maybe does not like this at all. We don't know yet. So I think we need to stay on our toes. The good news is though that the first generation of vaccines worked pretty well. So
that is really interesting. It's like that the existing immune response, right, based on prior infections, is actually gives people a natural boost.
Yeah.
We hadn't thought about that.
I've got a question too, if I may.
Yes.
Any idea why there's such a discrepancy from San Diego, where myself, you, and Dietrich live, where I think we have a couple thousand cases a day, and Austria, where there's 58,000 cases, I think, today? What's the difference? Why there's so many more cases in Austria compared to San Diego?
I mean, one easy difference would be the vaccination rate is much higher here, right? So it could have a difference. Children here are vaccinated all the way down to the age of 5. So also we know that this is almost a seasonal occurrence of the coronavirus, right? So it's hot, it's warmer here. It's I know back in Austria, you're preparing for spring. Here we already have warm temperatures. That could be a difference. And other than that, I don't know. So certainly the pre-existing immunity is not specific to Southern California. We see 50% of people here, but we measured on the East Coast, 50% here, we measured in Italy, 50% there in the Netherlands. So that's not the difference. People have equally levels of pre-existing immunity almost around the globe, but everywhere we look. Yeah.
Daniela, what is the story with the Omicron 2 virus that is circulating in China?
Yeah, so that's exactly one of these variants that occurred that seems to be even more infectious. That doesn't mean it's more lethal or inducing more disease, so that remains to be seen. And what we still don't know at all is how does our immune response recognize this new virus, right? And as I said, we still don't know how high does the wall need to be. Does it need to, do I need to boost because it's this high? Or is what we have good enough? We don't know yet. So that's what I'm saying. As long as we allow the virus to circulate, we will always have to asked these questions, because we're giving it a chance to mutate and produce more variants.
But there's also always the discussion around herd immunity, right? They are more and more transmissible. So at one point, we must reach a 70% infection rate across the world, and then what? And then does it become the regular flu and people walk away from it?
Well, that's a question of time. We really don't know. Yeah, I mean, if you think what happened to the other common cold viruses that have been circulating, that's pretty much what it happened. But what it takes to get there, the threshold, we will see. Yeah.
Elisa.
Yeah. Hi, Elisa Athofer here. I'm going to ask the Alex Jones question. So now that you've worked with COVID for a while and you've worked with so many other viruses and probably know very well, you know, which viruses mutate and how do they mutate, what tendencies do they have? What do you think about the theory that the virus originated in a lab and is man-made?
I mean, everything that we know so far does not point to that. So there seems to be like, you know, and this is not my my expertise, but like from what is published and what is out there, it doesn't look like it's originated in a lab. So I certainly don't believe that, but like as I said, it's also not my expertise.
And it's almost, what difference does it make at this point? We have the virus, we need to deal with it. It's like climate change, right? But whatever the reason is, it's happening. So we have to react.
Yeah, I think going forward, we should also like learn from like what's happening, because it's a matter of time until the next pandemic is coming. So we better be prepared, I think. So.
Is it true, I'm like hogging this, I'm sorry about that, is that the remnants of the Spanish flu are still sort of with us in terms of the new flu viruses that we're exposed to?
I mean, certainly, so we have, that's why I'm saying this is not a novel concept, that pre-existing immunity against one virus can influence infection with the other one. And this Spanish flu actually has had an effect. I'm sure you all, so influenza viruses have always been recombining with different proteins, right? And there has been a case of, I'm sure you heard about the swine flu, H1N1, where people have already had previous immunity, because typically after influenza, like very young people or very old people get very sick. In this case, the middle-aged people did, right? So that's because like older people had still like immune memory from this other virus. So yeah. So if that's what you're referring to, reminiscence, yes, there is some people that have immune memory against that. Yes.
I'm afraid the soda bread is.
More questions? Anyone in the audience?
I'm gonna have to cook it longer.
Those on the screen? Dietrich?
Oh, just wanted to again to say thank you for this wonderful talk.
Yeah,
It's you know, fantastic to see, you know, the progress that has been made in the field, you know, by Austrians, you know, all over the world. So I think this is really, you know, exciting to see. This is, you know, it's a good example of a topic that's you know, is on top of all of our mind with all of the areas that we are working in. All the way from immunology to, you know, public policy. So yeah, thanks for this great talk.
Thank you so much for having me.
and congratulations again on your award. It's
Thank you.
Mm-hmm.
Thank you so much. Then we close it out. If there are no more questions, then we thank you.
All right.
And because we are Austrians, we need to close on time. We can't be late. Bye-bye.
Right. Bye-bye. Thank you.
Thank you so much.
Cheers.